Okay, good afternoon, everyone. Thank you for coming. We welcome you at Memorial Sloan-Kettering. And for the beginning, I would like to ask Tanya Statman to give you a few words, and then I will continue with the schedule. We have a packed schedule today with a lot of good speakers and experts in the field. A program is listed there. We'll try to cover from very basic research, mouse models of GIST and imatinib-resistant GIST. We'll continue with update in pediatric and wild-type GIST. Research, as well as our clinical colleagues will give you the latest on the clinical management on advanced and the refractory GIST with the latest clinical trials available here at MSKCC. So Tanya, please.
Hi, everyone. Of course, many of you know me. My name is Tanya Statman. I'm a GIST patient, and I'm also the chairperson of GIST Cancer Research Fund. First, I want to thank Carolyn for organizing and putting the presentation together for us. So thank you, Carolyn, for all that you do. I want to thank all of you who have been here before, and I'm glad that I see you all here. So that means that you're doing well. And I'm not so happy to see some new faces here, but on the other hand, I am glad to see you here because you are in the right facility where you have a lot of the doctors that know exactly what GIST is, and they have a lot of knowledge about GIST. That's one thing that all the phone calls and the email and submissions that I get are from people that actually have been diagnosed with GIST, but unfortunately have not been able to be treated by the doctors that have any knowledge of the protocol treatment for GIST. So a lot of people and a lot of patients with GIST have been told to go on the internet and search for GIST. Or just sent home and they were told that they don't need to follow up with anything. So in many respects, I'm very grateful to many doctors at Memorial Sloan-Kettering, of course, that they have been very gracious with their time to helping me help patients, like Rhonda, Mateo, Christina, and even Bill. I think I sent you a couple of emails. And I even had one that was from Mongolia, I think, that Ron, you helped me with that, right? So the point in the matter is that the research for GIST is so pertinent that we follow through and we support, because number one, the one thing you have to know about GIST Cancer Research Fund is that we basically don't get compensated for the work that we do. We don't have an office per se. We work out of my bedroom. It's my office, and I don't have a secretary. And so all the work that's being done is being done by the directors and by the GIST Cancer Research Fund people that are basically like myself, a GIST patient, a caregiver, or many of them are also people that have lost a loved one to GIST. We have to understand the seriousness of the fundraising. I created that at a time when I was told to go home and get my life in order because I wasn't going to live more than that year. But this time, this is not about me creating GIST Cancer Research Fund. It's about the fact that all of us here, we are GIST Cancer Research Fund. Because all of you, hi Aura, how are you? Because all of us that are here and all of us that I know are present, and I know each and every one of you, is the ones that have supported and continue supporting GIST Cancer Research Fund. Why is that so important? It is important because every dollar that we fundraise goes directly to the researchers that do research for GIST. It's very hard for you to imagine that the doctor will have to sit and write grants, to get grants from the government. That is something that is very hard to get. The doctors, the researchers shouldn't even have to do that. Unfortunately, sometimes they do, but they don't get any kind of grants for GIST. So we have to do it privately. And the thing about it is that we don't get enough recognition. If you go into any area, in a supermarket outside on First Avenue, and you say GIST, nobody would know actually what GIST is. We're not the status cancer, and therefore we have to make all the efforts to make sure that the research for GIST continues. GIST is not as simple cancer as we might think. It's complicated. For many things that we knew in the beginning, many things have now been researched and researched and researched. It's not only that you have known what exon mutation you are, but now we're looking at different things. You have different types of exon mutation. You have not only GIST, but you have wild GIST, pediatric GIST. There's so many different things, and also we're working on so many different clinical trials with different drugs. Therefore, that's why we need to be able to support directly the research that's being done by Christina Antonescu, who is the pathologist and researcher for GIST, as well as Dr. Ron DeMatteo, who does the immunization research for GIST, and all the rest of the doctors. You know, we have Dr. Bill Tabb, Dr. Pinching, that works directly with GIST and GIST patients, and of course it's all from Peter Besman, Dr. Peter Besman's laboratory, that he actually started many things that most of us don't really know and how important they were towards GIST. So the point in the matter is, I'm standing before you because you're all part of this. And if we're not going to help raising funds, nobody will. I know how it is, the frustration of many people that I've talked on the phone or tried to call and get companies to be helpful to us, to actually raise their hands and help us with the research, with the funding. But unfortunately, being that GIST does not have that big notoriety, and because I, as a chairperson, I don't have the big notoriety, many doors have been slammed, many phone calls have been slammed, and here I was completely heartbroken because I take it personally, and basically we didn't get too much going. We didn't get too much help. So therefore, it's up to us to do it. So I really, not only that myself as a cancer patient, I'm talking to you, but I'm talking to you as a human being to another, because now you know what it's like to deal and have to walk that path with GIST. So I need you to understand that we need to fundraise as much as we can. And not only by just sometimes writing a check, which we do appreciate it. If it's $5 or $10, we appreciate it totally. But we need to bring in the families. We need to bring in the friends, anyone that you might think you might know, and totally out loud talk about GIST and getting people involved. Because that is the most important thing, because you never know who you're talking to. They might be able to help us in a different way. So I appreciate, all of you coming here. I know all of you are ready to hear so much about the research that's being done and hear from all the doctors. So I thank you so much. I am very humbled to be surrounded by all of you. I'm very humbled to be of some help to maybe a few, and I'm trying together with our directors on board to do our best to be there for everybody and to help all the researchers. We're helping not only Memorial Sloan-Kettering, we're helping at our facilities, but the point is that we have the loyalty to those who have and are doing research for GIST. So with that said and done, I think that we all need to do a little bit more and open our way of the way we're seeing things in that box and see outside the box. So I thank you so much, and just remember that we couldn't do what we do if it wasn't for you. So my husband is kind of saying, to Eleanor, okay. So anyway, and okay, so this is another. I wanted you to I want to bring something up. Eleanor, hey, wave to everybody. Okay. Eleanor is a GIST patient, and I just wanted to give her the recognition. And I'll tell you why. It's very important for you to know. Eleanor has been a GIST patient that's been going through the whole 9 yards of different drugs. And now she's on a new clinical trial. But I've known Eleanor from day one, and sometimes even from the hospital bed. She's telling me, make sure that you put on the calendar, make this or make that, because I'm going to have the event for fundraising. You are unbelievable. You are totally an awesome person, I have to tell you.
You have all of you. It takes a whole tribe.
Yeah, I know it's a whole tribe, but you know, it just takes one to bring the tribe together. So you are the one that does. So I really thank you for all that you do for GIST and for all of us. It benefits all of us. So thank you very much, and we're going to just turn to whoever wants to make the first speech.
Thank you, Tanya, for the wonderful words, and we appreciate so much the efforts and hard work that you do in raising so many precious funds for our work. We couldn't do without your help, and you will see from the amazing data that we have accumulated so far, and we are expanding every year with more people, more scientists, researchers being interested in this disease. So we all thank you, and we are happy to host you today here. Okay, moving on to human research. I think what we learned from Benedict's talk is that the success story that we all know of imatinib in GIST is really due to the presence of a single point mutation or deletion that occurs in the KIT gene, which results in the oncogenic activation of the KIT receptor, which in turn then makes the tumor highly sensitive to KIT inhibitors such as imatinib. And obviously this success story can be summarized in one slide shown here. And the good news is that most adult GIST patients fit in this scenario. About 80% of adult GIST will have an identifiable KIT mutation, and sometimes in a PDGFR alpha gene that also may be responsive to imatinib. However, this is not the case with wild-type GIST. Wild-type GIST do not show activating mutation in KIT or PDGFR alpha or BRAF, and comprises about 10% of adult GIST and most of pediatric and syndromic GIST that are listed here. And despite the fact that these tumors lack an KIT mutation, they express a lot of total KIT protein as shown here, as well as showing KIT biochemical activation by Western blotting. And if you look at gene expression profiling, the pediatric GIST as well as the wild-type adult GIST show very high level of KIT, actually indistinguishable from the KIT mutant GIST. Based on this finding, we cannot really distinguish between different subsets. So our lab has really took advantage of the latest technology of next generation sequencing, and we tried to use next generation sequencing in trying to identify potential new mutations in pediatric GIST and wild-type adult GIST. And we tested 7 cases in which DNA was available from both tumor and normal. And in order to exclude a platform bias, we analyzed these tumors on 2 different platforms, Illumina and Solid, and used very high coverage to identify novel mutations. And to our disappointment, only 1 of these 7 cases, which in fact represented a young adult wild-type gastric GIST, had a mutation, and that mutation was a germline mutation in SDHA, or succinate dehydrogenase A. Germline mutation meaning that that particular mutation was present in both tumor and normal tissue such as blood. And this patient, in fact, was refractory to multiple tyrosine kinase inhibitors listed here. So going back to the 6 cases of pediatric wild-type GIST, where the whole exome sequencing failed to identify new mutations. In addition, we performed a different type of sequencing in which we looked at RNA expression, and in the end, there were no identifiable genetic abnormalities. There were no somatic mutations and no chromosomal translocations that could be found in this subset of wild-type pediatric GIST. So what may be the cause? If there's no genetic abnormality in these tumors, then what is the abnormality that triggers tumor formation? So we all know that most pediatric wild-type GIST and a subset of adult wild-type GIST show loss of SDHB or succinate dehydrogenase beta expression, which is retained in most adults with KIT mutations. So what is SDHB? Well, SDHB is part of a complex, enzymatic complex. This slide is very busy, but just to make the point that the SDH, where there is a deficiency in pediatric GIST, actually is a very important step in the respiratory Krebs cycle in the mitochondria. So if the SDH complex doesn't work, the result is the accumulation of succinate. And it has become clear recently that a number of cancers develop, in fact, by toxic accumulation of certain oncometabolites, including succinate. And this can occur not only in pediatric GIST, but in other tumors in which SDH can be mutated or being abnormal, such as paragangliomas. So the fact that succinate accumulates in toxic amounts in the cytoplasm of the GIST cells, that triggers abnormality in methylation and abnormality in general in the epigenetics, including the stabilization of HIF-1 alpha. So thinking about this hypothesis that we don't have a genetic abnormality, and in fact what triggers pediatric GIST may be an abnormality in epigenetics, we started a pilot study in which again we explored the methylation or DNA methylation profile in pediatric GIST. And again, this is quite complicated slides, but the point here is that the SDHB deficient GIST from a methylation standpoint is extremely different. It's like a black and white difference between from all the other GIST types as well as other soft tissue sarcomas. And the difference is that you have global methylation. So DNA, the pediatric GIST DNA shows hypermethylations throughout at the promoter regions and CPG islands. And this is just another way of looking at it. We call it a volcano plot just to emphasize the massive amount of the methylation probes that show high expression. And if the promoter of a certain gene is methylated, then the result will be repression or decrease of expression of those particular genes. And here are just 2 examples of genes that in fact go down as a result of hypermethylation in the pediatric GIST. So what is the next phenomenon? Well, once the DNA becomes hypermethylated, the next step would be to recruit another member of that is important in histone methylation, which is HDAC. So DNA methylation triggers forming these HDAC complexes, which represents the histone deacetylases, and in turn produces more hypermethylation of the DNA. And the end result is that you have a stable repression of transcription of multiple genes across the board. And indeed, in pediatric GIST, the HDAC, for example, was highly upregulated in keeping with this hypothesis. And furthermore, one thing that we found to be significantly upregulated in pediatric GIST was the upregulation of a subset of the forkhead box proteins, 3 of them being illustrated here, and the same phenomenon occurs if the FOXP are up, then they just recruit more and more of this HDAC, and you have increased acetylation and increased methylation, and in the end, the repression of gene transcription. So how can we link all this epigenetic changes with the GIST? So GIST, pediatric GIST, although we don't have mutations, they have increased KIT expression, they have increased ETV1 expression, and now we learn through this work that they have increased FOXP1 as a result of the hypermethylation I just showed you. So one scenario that we are actively following in the lab is the hypermethylation of this particular gene, the EID3, which is an inhibitor of a master regulator of histone acetylase transferases, as well as a master coactivator of endogenous transcription. So this particular coactivator results in transcriptional activation of KIT, ETV1, and all these FOXP members. And we think that may be one key in which we can show that the presence of the SDH, the oncometabolite, increases methylation and then in turn increases the activation of this coactivator, and it triggers possibly KIT formation, GIST formation in interstitial cell of Cajal. So this is the story that we have right now. It's obviously to be followed. Many experiments are ongoing in the lab to prove that's the case. And hopefully next year we'll have a complete story that we could link epigenetic abnormality with formation of SDHB deficient GIST. A few words about imatinib resistance. So we have a different project in the lab looking at additional and alternative mechanism of resistance. And we took the same approach, looking at the next generation sequencing in a 10 imatinib resistant GIST in which we had DNA available from matched normal and tumor. We try to select mostly imatinib resistant tumor that failed imatinib after a period of good response, so looking at mechanisms of secondary resistance. And of course, we selected mostly patients where there were no secondary KIT mutations so no identifiable mechanism of resistance. And the emerging mechanism here, which is in a way similar with what we are describing or finding in pediatric GIST, is that a number of these imatinib resistant samples show mutations in chromatin modifiers genes. So again, in epigenetics, either histone demethylases or histone methyltransferases. So we are now trying to screen additional cases to find the exact incidence of these mutations in imatinib or sunitinib or other combination therapy resistant samples. This work we are doing in collaboration with Ping Chi, and obviously this is a completely new field. We think that epigenetics alteration may have a significant role in imatinib resistance. So we will hear Dr. Tapp talking a little bit later about new clinical trials. And one of the new trial that I think will open soon at Memorial will include a combination of imatinib plus FGFR inhibitor. So based on On this trial, we looked at our imatinib-resistant samples that we had on the gene expression array, and we wanted to see the expression of some of these receptor tyrosine kinases to see if we can predict, in fact, patients that have upregulation of the FGFR1, FGFR2, and potentially select a subset of these patients that we may think have a better chance to respond on this combination therapy. As you probably know, imatinib-resistant GIST, the mechanism of resistance are typically KIT-dependent. So once the tumors fail responding, we typically have overactivation of the KIT protein as shown here. The tumors start proliferating very fast. However, we also know that the imatinib-resistant GIST is a very heterogeneous disease. So we may not be surprised if additional feedback loops are possible with upregulation of other receptor tyrosine kinases, such as FGFR1, PDGFR alpha, and so forth. And you can see here that a subset of the imatinib-resistant GIST have upregulation of FGFR1, and some of them significant upregulation of PDGFR alpha when you compare with the imatinib-sensitive disease. So we are continuously mining and using the genomic data that we have and trying to integrate with all the ongoing clinical trials or the new clinical trials that will be available in order to identify and tune the selection of patients or subsets, molecular subsets, as much as possible. So I think this is my last slide. I obviously want to acknowledge many people here. My mentor, Peter Besmer, without his help, I could have never started working on just Ping Chi, who's bringing to the table a completely different expertise. We have a lot to learn from her epigenetics knowledge. Ron, of course, always a good friend, and we learn a lot from his immunotherapy trials. And of course, my dedicated lab, Peter Sang, who is doing all the bioinformatic work, and Manu Tirunagari, who's doing all the in vitro cell culture work. I couldn't do without such a talented and dedicated lab. And of course, my other clinical colleagues, and a big thank you for all your support. Thank you.
I very much appreciate the opportunity to just spend a few minutes talking to you. I always feel that the patient-centric conferences and meetings are probably the most important that we do. Why? Because I think to a person, all of us are here because of you, and it's the patients that give us the ability to go to work every day and try to find meaning in work so we can bring the work back to the clinic. So I think it's really important for us to communicate what we're trying to do and how that could impact patient care. So thank you all for being here. I also, having been in this business a long period of time, know that we have to know our roles, and I think my role today is going to be more of the setup man for some of the excellent research you're going to hear. So I thought what I'd like to do before we got into some of the clinical trials is just bring everyone to the same page about what GIST is and why is it a disease that we struggle with, but also why is it a disease that we've seen so many advances in. And so GIST originally was known as a leiomyosarcoma of the GI tract. So these were actually thought to be tumors of smooth muscle, so muscle cells of the GI tract, and they were rare. There was only about 400 people diagnosed with these tumors in the United States each year. And surgery was actually the best treatment we had for these tumors. For people who actually had metastatic disease, meaning more than one spot, or they recurred after surgery, this was a fatal disease. So people would die often within a year or 16 months. And why was that? Well, because these tumors didn't respond to any of the therapies we normally use in cancer. It didn't respond to radiation. It didn't respond to chemotherapy. So it was a very tough disease to have. And it was a disease, though, that was destined to become one of the hallmarks of the cancers that we actually treat. And I think the history of this is very important. It started from some very interesting observations that these cells weren't actually cells of muscle, but rather they were cells of the interstitial cells of Cajal. And so that's something that we always throw around, the ICCs. And what are they? So Cajal was actually, I believe, a Spanish pathologist, and this is his picture up here on a stamp. And he actually identified the interstitial cells of Cajal. And these are cells that actually sit within the muscle layer of our bowel. So if you can picture this, a tube where the inside of the tube is up here, and these are the muscles. In between these muscle layers are actually these cells known as the interstitial cells of Cajal. And we call them the pacemaker cells in a way of the GI tract. And what does that mean? Well, we know when we digest food, food has to be ushered out throughout the whole GI tract for actually it to be digested. And so the bowels can actually push the food down. And it's these cells that kind of pass the neurological impulses and tell the bowel when to do that. So a very unique cell in the system. But that observation was really important because we know when these cells actually develop from a stem cell and then they turn into the mature cell, they really rely on this receptor known as KIT that we always talk about. So that was the first clue that maybe this receptor, which is important in the development of the cells of which the cancer comes from, maybe they're important in the cancer. And that was critical. And what is KIT before we get into it? So KIT, we know, is this transmembrane type of receptor. And what is that? I tell all my patients it's like the old rabbit ears on a TV. So they basically sit on top of the cell, and it receives signals from the cell. And when it receives those signals, it can turn on different things within the cell. And normally what happens is when a cell is in a healthy environment and the body wants those cells to grow and duplicate, it'll send it signals. The antenna will pick up those signals. And then a series of events will happen within the cell that then tells the cell to grow. And normally this is a system that can be turned on or off, just like we can turn a light switch on or off. The question then became is how is this receptor, if so, involved in the cancer? And it was interesting because if you understand this receptor, you actually know there's a hereditary condition that this receptor is not in people. And when people are born without this receptor, they have a very characteristic way that they look. They have a disease that's known as piebaldism. And so this is an interesting disease, and it's named piebaldism because it's named after the magpie and the bald eagle, both of which have these kind of characteristic white patches on their head. And why is this happen is because this receptor is so important in the development of pigment in our melanocytes, right? But clearly our patients don't look like this. So it was thought that this isn't probably something where the receptor is absent. Interestingly, if any of you have been on certain drugs that knock out this receptor, your hair turns white, right? And it's the same concept. But so it wasn't that this receptor was active or absent. So the next logical step was that this receptor was probably always working, so always turned on. And that's what we know is now happening in GIST. So instead of having this receptor on the cell that's waiting for the body to tell it to grow, this receptor is always turned on, like the light always turned on. So these cells always think they're in a mode to grow. We need to grow. We need to grow. And that's what actually causes these tumors to develop. This was some of the work done by a group at the University of Kyoto in Japan that actually demonstrated this. And this was in 1998. And it was shortly after this, when they demonstrated that this receptor is always turned on, that it gave clinicians and a lot of thoughtful people a way to potentially target that receptor and turn off that receptor. And that's where imatinib or Gleevec came in. This was actually a drug that was being used for CML, and it was being used under a different principle. But it could actually sit on the top of this receptor over here, on the bottom of this receptor over here, and although the receptor thinks it needs to signal, it prevented these signals from coming down into the cell. And this was a remarkable discovery because almost overnight, once people started using these types of drugs, what we found were tremendous responses in patients. And this was the first patient who tried it. in 2001. And what you can see is they had tumors all throughout the liver. They had progressed on chemotherapy, radiation, and then all of a sudden we were seeing tremendous responses. So that allowed for a larger study. This was a phase 2 study that actually looked at how patients did on this drug. And what you can see is in people with really advanced tumors, instead of people dying early on within 12 to 6 months, people were actually living. And so this was a major breakthrough in medicine, and it happened in GIST. We know that people can have tremendous responses in PET scans. This is what a PET scan looks like, where you have this activity in the tumor, it gets shut down. And on a CT scan, when you have a big tumor, the tumor can shrink. So this type of study led to 2 larger studies, one in the United States and then one in Europe, and this led to FDA approval for the drug, and in the Europe, EMA approval for the drug. And everyone was really excited about it. What was also interesting is when you actually looked at who was responding to the medication. And one of the things that you can actually do is look at this receptor and find exactly where there are mutations in this receptor to always turn it on. And that's what we talk about when we talk about exon 9. That's when a mutation happens in this area right outside of the cell. The most common is an exon 11 mutation, and that's the area right under the cell membrane. And it's just that simple mutation that you heard before that can actually turn that receptor on. But we now know that in people who have certain mutations, like an exon 11 mutation, they do better on imatinib than people who may have an exon 9 mutation. So it was critical not only to demonstrate that you need the mutation, but also it's important to know where that mutation is. And now this has become common talk amongst patients. So patients come in and they'll introduce themselves as having GIST, but I have an exon 9 mutation. How is that going to affect my treatment? But this is what we're talking about, just subtle places within a receptor that's actually turning that receptor on. So what happens, unfortunately, is people who have advanced disease, although they have tremendous responses to this drug is not curing patients. There are some people that can remain on this drug for 10, 12 years and longer, but there are other people that after just a few years, the tumors start to grow. And we now are beginning to understand why. And there's a whole list of why that resistance develops, some of which you'll hear today. The most common is what we call a secondary mutation, where being on the drug actually selects for cells that have mutations in other areas of this receptor. And what does that mean? I explain this to a lot of my patients, is that this receptor and how Gleevec works is really like a lock and key. So Gleevec can fit into that lock just like a key does and can actually shut off the signaling. But when these other mutations begin to develop, they develop in areas in that lock that actually prevent the key from actually fitting in there. So it's almost like changing one of the prongs in a lock so the key can no longer fit there. So the drugs for those cells doesn't work. And then over time, those cells grow up into a tumor. It's also like spraying Raid on a group of roaches. If you kill 90% of them, but 10 of them or 10% are resistant to Raid, they're going to start to grow up. And over time, you'll have all of these cells or roaches that are actually resistant to the drug. So the other way, which we'll talk about too, is there are other types of signaling that the cell can acquire to begin to grow. So even though we can shut down one of the main pathways or receptors that the cell uses to grow, it can actually signal through other ways. And so these are all things that we're trying to do to actually allow people to do better on drug. Now, we were fortunate again because when we first started to see people who were becoming resistant to Gleevec, there was another drug around being developed in, this time, a kidney cancer called sunitinib. And sunitinib was different than imatinib in that it was a much smaller drug. So even though we started to see those resistant type of mutations where imatinib or Gleevec couldn't fit into that lock, sunitinib could. And because of that, we now use this as the second-line therapy for people once disease starts to grow on Gleevec. And this was the study that was run showing that when you give this drug, instead of doing nothing for patients, you can actually improve how they do. But unfortunately, sunitinib is not the answer for a large majority of patients because it only works on a subset of changes that happen within that lock. So there's a group of people that when they start that can actually do very well. But we also know that there's a group of people that sunitinib doesn't help that much. And we're beginning to to understand why. The problem is, that was kind of alluded to by Dr. Antonescu, is that once we start to see resistance develop on a drug, it's just not one type of resistance. It's just not one type of mutations. And we now know that if we would sample different tumors in the body of a person with GIST, when it starts to become resistant to a drug, we actually see many different locks forming. So there are many different types of mutations that happen. And this becomes very difficult for us clinically. So what would happen is that if we would start a second drug, like sunitinib, it may be able to control certain tumors, but then other tumors can begin to grow. So what does that do? We then switch to another type of drug that may be able to control these tumors, but then the original ones begin to grow. So we kind of liken this to that old game of whack-a-mole in the field, that we're really reacting to what the tumor is doing to us, as opposed to trying to get one step ahead of the tumor. And that's one of the points that we're trying to do with the research, is how do we even prevent getting into this state where we're now reacting to the tumors. But it's also a problem for us, because what we find is the more medications that we put patients on, the actually shorter time that they respond to those medications. And that's just because the tumors are developing more ways to overcome the treatments that we're giving them. So now one of the focuses that we actually have here at Memorial is to try to find strategies to overcome this resistance that's developing. And one of the ways is actually to look at drugs that can work in spite of all of these mutations. And this is a very promising field, because we can now look exactly at where those mutations are in this lock and figure out how to engineer drugs that can actually develop into that. We're looking at a lot of combination therapies, because the thought is that using Gleevec may still offer patients a benefit. And if we can add a second drug to that, we can really add power to Gleevec by preventing these cells from developing these other ways to signal and grow. One of the important things that I'll touch on briefly is we're looking at adding these therapies early on and when a patient first gets diagnosed. So if we can bring these treatments early on, maybe we can cure patients instead of allowing them to be on therapies when all of these types of mutations develop. And then we're looking into other targets, and Dr. DiMatteo is going to talk a little bit about the immune system. So this is just an example of a drug known as ponatinib. And ponatinib is a drug that's been getting a lot of press lately, because it was thought that it could overcome a lot of different types of resistant mutations. And it's actually proof of principle to the concept of, can we design a drug that can fit into every lock that happens within the body. There was a lot of promise of this, but this drug is stalled right now because it had bad side effects. But I think what we're beginning to see is actually newer drugs like this really push into the market. And what's the power of some of the research being done here is that some of the labs have really been able to develop all of these mutations within their labs. So when these new drugs come, we can quickly test them to actually say, is this something that can help a wide variety of people? The other thing which people are working on is really trying to figure out how to block all of the different pathways that these cells can actually develop. And so what could happen is, once this receptor really starts to signal, we block it. It figures out how to overcome that blockade by using other signaling areas within the cell. So it's basically doing the same thing that it started out, but through a different pathway. So now the thought is actually to combine two drugs. And this is work done by one of our collaborators actually in Belgium. And they started a clinical trial for people who have been on imatinib, sunitinib, and other drugs to really see if they can now use two drugs. They can block all of the resistance to develop. What we're really excited about is some of the work that you're going to hear from in a little while from Dr. Chi and Dr. DiMatteo. And I just wanted to introduce this because Dr. Chi, we believe, has found another critical aspect in the signaling and development of GIST. And the thought is that this other element, which is known as ETV1, really cooperates with this receptor KIT in developing GIST tumors. And the thought is that if we can block both KIT and this ETV1, we can really not only shut down GIST, but we can actually cause these cells to die. And so that's an important concept, because instead of giving it a medicine like imatinib, which actually controls things, we're actually going to try to kill these tumors. And this was something that took a long time to develop and a long time to convince our pharmaceutical colleagues to actually support. But the way we're running this clinical trial now is actually in patients who have newly diagnosed GIST. So this is changing the mantra of just saying if you're newly diagnosed with GIST, you should be put on imatinib. What we actually want to do is place people on imatinib and a second drug that targets this ETV1 protein, because we feel this is the time where these tumors have not yet developed all of these types of resistant mechanisms, and we can really begin to cure patients of their disease. So it's a proof of principle trial. It actually just started maybe about 2 to 3 months ago. But the key is we actually have to bring people back from the community who now have GIST, they've been diagnosed, and they're going to be treated by imatinib by their local oncologist, to say, we're hoping we have a better way to do this. So let's bring you back to the academic centers and actually try this combination. The other thing, like Dr. Antonescu spoke about, is looking for other ways these GIST cells actually can get around the use of imatinib. And some of the ways they do that is they actually bring other receptors to the surface, almost replacing or working together with that KIT receptor. One of which we're very interested in is this receptor known as FGF, which is known as the fibroblast growth factor. And we think that this is just another receptor that the cell brings to the surface to allow the cell to grow. And similar to the clinical trial in which we're running with Dr. Chi with a drug known as a MEK inhibitor, in about 3 months we're going to be starting this clinical trial, which is actually with an FGF inhibitor. And why is this important? And people ask, well, why do you have 2 clinical trials in similar patient populations? Because we believe that how GIST works is different for different people. So we can't just commit to one type of way to study and treat this disease, but we have to be as crafty as the disease and understand that if you line up 10 people with GIST, you're probably going to have 10 different variants of the disease. So we have to really figure out how to individualize the treatment strategies. And this is one way to do this, to look at the patient's tumors and to begin to say, what type of treatment do you need to be on? Dr. DiMatteo is going to talk a little bit about immunotherapies. And immunotherapies are critical because they offer us different hope than some of these other drugs that we've been using. This is data actually from melanoma. And melanoma, like GIST, has a very specific mutation. And when you use a drug like Gleevec in melanoma, what you find is that people can have really nice responses early on. But the problem is those responses are not long-lasting. Just like GIST, resistant develops. But in melanoma, they also have therapies that are immunotherapies. And what you find is that these therapies do not work quickly. But when they work, and in the patients they do work, they can actually cure patients. So what we're trying to do, and we have a clinical trial now, is to combine a small molecule like imatinib with immunotherapy. Because the hope is that we can get a quick response for our patients and allow the immunotherapy work where we can actually begin to have long-lasting types of responses. So the combination of these 2 therapies are also going to be critical. And so that trial, if Dr. Carvajal comes later, he will speak about. But to me, I think this is the most important thing that we're trying to develop at Memorial, and that's an individualized treatment strategy for our patients. As I said, everyone with GIST has different variants of the same disease. What we have to start to do is understand those variations so that when people come into our clinics, we could say, this is the right drug for you. And that's something that is one of our goals of the GIST program here. But what's critical and humbling is we can't just say there's one way to do that. What we need to do, and I hope what you understand today, is that we're developing strategies to target and treat GIST that actually develop in all different aspects of science and clinical care. So we have to look at immunotherapies in the immune systems. Like Christina spoke today, we have to figure out how proteins work and how proteins develop. We have to look into the genetic aspects of that. And then we have to understand the heterogeneity that comes with all of our patients and say, how do we match the 2? So in my opinion, we have a wonderful center here because we have really dedicated physicians and scientists who work in all different aspects of research. and they're trying to bring all of those aspects together for our patients. And that's an important aspect of this meeting today, and one of the take-home messages that I wanted you to get. So that's all. Thank you.
I really appreciate the opportunity to update you on our recent laboratory work. And on top of that, I really appreciate all of your support for JISC research, because without you, and we wouldn't have been here. So let me begin with, many of you know that actually predate the kit discovery, just under the microscope looks very differently, and they are often misdiagnosed. And now we know they come from the same cells, cells of precursors, the interstitial cells of Cajal, and the majority of them, about 95% of them, would highly express KIT, a receptor tyrosine kinase, on their cells, on their surface. And in 1998, Hirota and his colleagues had made a landmark discovery, as Dr. Tapp alluded earlier, that they have discovered that the KIT and PDGFR alpha mutation are present in GIST, and these are the mutations that are actively driving the GIST tumorigenesis. And with these, and now we know about 75 to 80% of the GISTs will have mutations in the KIT gene, and about 5 to 10% of them will have mutations, activating mutations in PDGFR alpha, and about 1% in BRAF. And this has really revolutionized the clinical management of GIST. And they, with the advent of Gleevec, and as you can see, it significantly extends the overall survival as compared to the historical control. This is one of the phase 3 European studies. However, just Gleevec is, even though it's very effective, it's not good enough. If you look at the progression-free survival, you can see these curves never flatten out, which means that with Gleevec, it rarely cures the patient. It does slow down the disease progression, but eventually the majority of the patients will progress. And for two reasons, we think. As Dr. Tapp mentioned, so one of the most important reasons is the development of immune resistance. This is mostly secondary mutations, and also tumor persistence is one of the other reasons in some of these other genotypes, suggesting that the Gleevec just doesn't work very well. It probably suppresses tumor growth, but doesn't cause cell death in these tumors. So we need to. I think the current need for GIST management is really twofold. One is that we need to develop effective therapies, identify novel therapeutic targets to overcome immune resistance. And two is that we need to develop more effective first-line therapy, something more effective than Gleevec alone. So while I was a fellow, I thought a lot of the focus of GIST research has been focused on the upstream signaling pathways. And given my background training in the chromatin biology, I thought to look a little bit further in the nucleus. And what we found is that we found ETV1, a transcription factor. Usually a transcription factor turns on genes or turns off genes. It's significantly highly expressed in GIST compared to many other different cancer types. This is the Z-score on the y-axis. So this actually plays, this is the gene expression level. It actually plays the ETV1 expression level at the top 90-95% range. And if you look at the different locations of GIST, ETV1 is significantly highly expressed in both of the GIST coming from the stomach or the small intestine. And ETV1 is highly expressed regardless of the mutational status, whether they're KIT mutant or PDGFR alpha mutant or wild type GISTs. These are all sort of publicly available gene expression data that we mined. And the long story short, we did some more cell line work as well as mouse model work, and established that it turned out ETV1 is a master regulator for the GIST cells and its precursor ICCs. So without ETV1, despite the presence of the upstream signaling, there's no ICC in these mice. And in the presence of ETV1 and normal KIT signaling, you get a normal development of ICC in the GI tract. Those are the pacemaker cells. And in the presence of a KIT mutation, it causes a constitutive activation of the downstream signaling pathway, and which actually stabilizes the ETV1 protein and causes a hyperactive transcriptional program. And we think these two together actually drives part of the GIST pathogenesis. And we know from our mouse work that ETV1 is actually required for the GIST tumor progression and survival. And now, since the discovery of ETV1, we have done some further mouse work to really prove that ETV1 is a worthwhile target in vivo. First of all, we used the GIST mouse model that's developed by Peter Bessemer and his colleagues. And when we crossed the KIT mutant GIST mouse model with the ETV1 knockout mice, so what's shown here is that in the presence of ETV1, you see the typical human GIST-like cecal tumors. And in the absence of ETV1, and despite the presence of the KIT mutation, we no longer see these cecal masses. What we see is a relative preservation of the GI tract. And if you do immunostaining, this is a classical GIST tumor which stains highly for KIT and ETV1 in these tumor cells. And whereas in this case, we only see the ICC, part of the ICC that doesn't normally express ETV1, and the rest of the ICCs are gone, and we no longer see tumors. So this is to ablate ETV1 in germline, meaning this work really suggests to us that ETV1 is required for tumor initiation. And what happens if the tumor is already there, and then what happens if we ablate ETV1 or delete ETV1? So what's shown here is that if you ablate ETV1 after the tumor has already formed, the proliferation index, which is marked by Ki67, dramatically goes down. And we also see more tumor fibrosis with time. This suggests to us that even after tumor has already formed, ETV1 still matters. It still contributes to the tumor proliferation and survival. So with these, and on top of that, actually when we did some of the baseline immunostaining, which comes really as a surprise to us, is that when we ablated ETV1, we see a dramatic reduction of the KIT protein levels, and which is shown here in this immunofluorescence picture, as well as in these immunohistochemical stains. So this is the tumor cells. These are the untreated or corn oil sort of vehicle-treated tumors, and these are the tamoxifen-induced ETV1 ablated tumors. And we see a KIT expression reduction also in the ICC hyperplasia as well. And we see it at the protein level if we do just Western blot to blot for ETV1 and then for KIT level. And we see it at the transcript level. And then finally, we could perform some chromatin what we call chromatin immunoprecipitation, and to map where ETV1 is localized. And we see that ETV1 binds to the upstream enhancer region of the KIT locus, and it can actually directly regulate the KIT expression. So all of these actually suggest to us that ETV1 drives a transcriptional program, and one of the directed gene target is actually KIT itself. Based on these, we have a feed-forward model. What's shown here is that we think in GIST pathogenesis, in addition to the mutant KIT upstream, and which stabilizes the ETV1 protein, causes a hyperactivated transcriptional program, we think there is a positive feedback. The stabilized ETV1 can actually enhance the KIT gene expression, and this whole circle forms a feed-forward loop, and this is how ETV1 and mutant KIT drive GIST pathogenesis. And as you can see, or you can imagine if we could somehow target ETV1, we could also indirectly target the upstream mutant KIT and also the wild type allele oftentimes is present. So in summary, if we could target ETV1, we may be able to target not only the mutant KIT. We could not only target the GIST tumors that harbor mutant KIT, but also we may be able to target those GIST tumors that harbor PDGFR alpha or a wild type GISTs. Also, there is some evidence that part of the resistance mechanisms might be coming from these KIT-low stem cell or progenitor cell population, and it's being shown that ETV1 is actually required for these progenitor cell population survival. So by targeting ETV1, we might be able to get to this population as well. So how do you target ETV1? And this actually comes from some of the earlier work. We have noticed that if you treat the GIST cells with a MEK inhibitor that inhibits the MAP kinase pathway, you see the ETV1 protein level goes down very rapidly, even with a half an hour treatment. And if you treat them with the imatinib in an imatinib-sensitive cell line, you see the same thing. The ETV1 protein level goes down. And the ETV1 protein destruction is very well correlated with how well the cells respond to either a MEK inhibitor or imatinib. In this GIST cells, it's actually resistant to both, and then you see initial reduction of the ETV1 protein level, but reconstitution or restabilization of the ETV1 protein as you go to the 24 hours. And same thing with the imatinib treatment, and this is a cell line that's resistant to both. And in this GIST cell line, it's sensitive to imatinib, it's correlated with ETV1 protein destruction, but then it's resistant to MEK inhibitor because the MAP kinase pathway quickly reestablishes and then it stabilizes the ETV1. So based on these, we actually realized that with a MEK inhibitor, single agent is not going to work just because different GIST tumors may have different wiring. And one of the things we noticed was a surprise to us is that when we combined the two, the imatinib with the MEK inhibitor, we actually see a dramatic induction of the apoptosis, and even at a very low dose of imatinib. And this gives us a sense that perhaps the MEK inhibitors can augment the imatinib efficacy and by inducing more apoptosis. And perhaps this is due to the break of the feed-forward positive feedback circuit as I initially described. And when we treat the cells with the combination therapy, there is at least an additive effect when we used both of the imatinib. This is the imatinib alone at different doses, and this is imatinib alone for each of the color and in combination with different doses of MEK inhibitor. And the combination effect was even more dramatic when we treat the cells in vivo. So we xenografted these human GIST cells in vivo. As you can see, if you don't treat them or treat them with water, the tumor continues to grow. And with the single agent of imatinib, showing green, the tumor at the maximum or the maximum doses that the mouse could tolerate, the tumor only stays about the same. It does not shrink. And same thing with the MEK inhibitor alone. But when you combine the two, as you can see in black, purple, or orange, they actually, the tumor dramatically shrinks with different kinetics. They eventually achieve complete response. If we continue to treat them, and we have treated many of these mouses up to 6 months by now, and the tumors don't come back. And we've seen similar results in another GIST T1 cells, another imatinib sensitive cells. And when we did the Western blot, we realized that even with the thing with the single agent is that even at the maximum tolerated doses, we still have residual ETV1 levels. And this is completely abrogated when we combine the two drugs. And we can lower the doses of imatinib or MEK by half or a third, and we still could achieve complete responses. And this, what's shown here is just the histology. After 10 days of treatment of this sort of black line, and then you see a near complete response. This is at 10 days, but by 17 days, the tumor is gone. And lastly, when we treated the just genetically engineered mouse models, and we also see a more effective effect when we combine the imatinib with MEK inhibitor. And we see a similar effect in ETV1 destruction, and we see more fibrosis with the combination treatment. So taking all these together, and then really based on these preclinical data, and as Dr. Tapp mentioned earlier, we now have a phase 1b and 2 clinical trial. The phase 1b is actually primarily enrolling patients who has already progressed on imatinib, and I think we just completed, or we're very close to complete phase 1b, and the phase 2 trial will be focused on treatment-naive patients. And the goal is really to be able to develop more effective frontline therapy so that we could address the tumor persistence problem, and then we might be able to have a chance for cure. And this is more of detailed schematics of how we do the phase 1b. We start with a relatively lower dose of MEK 162, which is a MEK inhibitor in combination with the standard dose of imatinib. And then we can go down or go up based on the toxicity we see. And now we're actually expanding on this cohort, and then we recently just enrolled the last patient. And then for the phase 2, it's primarily for the imatinib naive patients. Will have a lead-in of imatinib, and then add the MEK inhibitor as they tolerate the imatinib, and then we'll have many correlative studies built in to really understand, why patients respond, why some of the patients don't respond, and hopefully we'll have some more tissue at the end to understand if they do develop resistance, why that is. So I couldn't really do all of this by myself, and really this is a teamwork, and that I like to thank my mentor. First of all, David Alice, who has taught me epigenetics, chromatin biology, and transcription regulation, and Bob Mackey, who really was my clinical mentor while I was a fellow here, and Jonathan Fletcher, who has given us all of the cell lines. But most importantly, I wanted to thank the service, Dr. Tapp as the service chief, and our surgeons, as well as Peter, who has been very supportive, giving us the mice and collaborating with us, and Christina, who has really been a great collaborator and instrumental on many aspects of things. And then the folks who did most of the work, and my other half, and also collaborator, Yu Chen, also a health faculty, and his mentor, and our boss, Charles. And of course, as I said at the very beginning, I really wanted to thank all of you for supporting just research. And without you, there is no motivation. And I think and these are my funding sources. Thank you.
For us to really make a difference in curing GIST, this is a team effort. We can't do this ourselves. The doctors, the scientists. It has to be a team effort with the patients. And so that's what this is all about here. I think you've gotten a sense so far of kind of the resources that we put into trying to figure out how to beat this disease. And a lot of the work that's been done, for instance, the work that Dr. Chi was talking about, is really how we use what we call targeted agents to fight cancer. And we've made a lot of progress with that, and a lot of that is due to the hard work that you guys have done, trying to help us develop drugs like imatinib and sunitinib and regorafenib and so forth. And these are all what we call targeted agents. But over the past few years, there's really been, I think, major advances in developing immunotherapies for cancer. Now we have several to fight various cancers, not just the targeted agents, but also immunotherapies And now a major effort has to be made to try to rationally combine these two types of different drugs. And one of the ways we're doing that is trying to combine the targeted agent dasatinib plus the immunotherapy called ipilimumab. And we're studying that in GIST. And I'll talk a little bit about kind of how this came about and what the trial is looking like and where we are so far. But again, with immunotherapy, we have to start thinking beyond the tumor cell. With the targeted agents, historically we said, well, the GIST tumor has this mutant-activated KIT protein. Let's just shut it off, and that's what Gleevec does. But there's much more to cancer besides that, and that's kind of what we call the tumor microenvironment. And so this is a little bit complicated, I guess. But here, here we have tumor cells. This is kind of a lymph node, and this just shows kind of the interaction between the tumor and the immune system, where your tumor has these proteins called antigens, and those are those yellow stars there. And those antigens have to be presented to the immune system. And that's done by these cells called dendritic cells. And then they go to the lymph nodes. And in the lymph nodes, an immune response is kind of triggered. And that's where it makes all these immune cells called T cells. And those T cells go on and fight the tumor. And there's some things that we can do to try to enhance this effect. And one is we can try to enhance the ability to present antigen to the immune system. And that's kind of one thing that we can do. I'm not going to talk about that, but that's one thing that we can do in terms of affecting the immune system. Two is we can promote the production of what we call protective T cell responses, or enhance the way that we can generate good T cells. And one of the drugs that can do that is this drug called ipilimumab, which I'll talk a little bit more about. And the last thing that we can do is we can overcome the immunosuppressive environment of the tumor. So it turns out that the tumor is always trying to your body's always trying to attack the tumor using your own immune system. But the tumor can actually evade that. And it kind of has these processes that can shut down or dampen the immune system. But there are things that we can do to overcome that. And so that's another thing that we can do as well. The new class of drugs, these immunotherapies, have been very, very, very effective, are what we call immunologic checkpoint blockade agents. And it turns out that if you look at one of these immune cells, these T cells, they're proteins on the surface. These are like antennae. And some of them, if they're turned on, turn on the immune system. That's a good thing. There are other antennae or receptors that if you activate them, they actually shut down the immune cell. And in terms of cancer, that's a bad thing. Ipilimumab is an antibody. So it's just a protein that binds onto this thing called CTLA4 on the immune cell. And it kind of blocks the ability of the immune cell to be shut down by that. It takes the brakes off the immune system. And by kind of using that antibody, ipilimumab, we actually hyperactivate the immune system. When we look at kind of the efficacy of these different treatments, they're really different. So if you look at how well these targeted therapies or small molecule drugs work, you can kind of see this curve like this. And I hate showing these curves, these survival curves. They're never as good as we want them to be. But that is what this is. It's not for GIST patients, but this is for melanoma patients using a targeted drug called vemurafenib. And what you can see here is everyone does well in the beginning with vemurafenib. Actually, almost all people do well for a while with vemurafenib. But over time, the cancer grows and grows, and patients start to not do well. And that's what happens with these targeted drugs. It works for a little bit, but it doesn't work forever. And so you get a curve that looks like that. And kind of some of the characteristics of small molecules in GIST and other tumors is that oftentimes they'll work very, very fast. Oftentimes, you'll get tumor shrinkage in most cases. But the tumors ultimately outsmart the treatment. They become resistant, and then the tumor will grow. But if you look at these immunotherapies, the curves are very, very different. This is ipilimumab in melanoma. And here what you see is the curve is completely different, actually. Unlike here, where most patients do well for a while, here, most patients don't do well. They just start just not doing well. But what you do see is a flattening of the curve, so that if you've done well by around 2 years, you continue to do well. And so this is kind of a characteristic of immunotherapy, where you have some delayed responses, so it takes a while before it works. You don't get a lot of tumor shrinkage sometimes, and you have this tail of the curve effect. And that is, if you make it out this far, you might actually be cured of your disease. Your immune system may be able to eradicate all the cancer. And so the question then is, how can we make those immune responses better? And it might be that if we combine some of these targeted agents, use these targeted agents with some of these immunotherapies, we actually may make that immunotherapy work better. And there are a lot of reasons why that might happen. Some of those are listed here. I don't know how much I should go through these, but for instance, one of these things is if we can shrink the tumor really quickly, then we can actually decrease the immunosuppression that tumor makes, so it lets the immunotherapy work better. Some of these therapies can also cause apoptosis or destruction of the tumor, and it actually allows more expression of these tumor antigens, that those stars that the immune system can recognize. It can also alter the antigens or those proteins that the tumors express, and that can make it more immunogenic or more kind of observable by the immune system. It can help enhance antigen presentation by those special cells called dendritic cells. It can modulate actually the immune cells themselves, right? Because these medicines are not just affecting the tumor, but they're also affecting your normal cells, including your immune cells. And that may be a good thing for the immune system. It may be a bad thing for the immune system. And it can also directly modulate or change the tumor microenvironment, the other cells, the non-cancer cells, non-immune cells in that tumor. And so what we really want to do is kind of combine the curve that we see with the small molecules, where we help a lot of people, at least for a little bit, with the curves that we see with immunotherapy, where the people who we help, we can help forever, and perhaps see survival curves like that. So that's our goal. It's challenging to do that, though. You can't just add two drugs together, because you add two drugs together, you get added toxicities, and you get other kind of unintended effects. For instance, some of the targeted therapies that we have, like the MEK inhibitors, for instance, that Dr. Chi was talking about, can actually suppress the immune response. So it can make the immune system weaker. So maybe we don't want to combine that with immunotherapy. Or if we're going to do that, we have to do that smartly. Maybe we have to sequence it or stagger it. Again, side effects might be an issue. One drug has side effects, drug 2 has side effects, you put them together. They might have a lot of side effects. And then again, the timing and sequencing of how we give these drugs, you really have to give a lot of thought to. You have to understand how these drugs work, not just on the tumor, but also on the immune system. And so at Memorial, we've done a lot of work clinically trying to figure this out. And so we have run or are running a series of trials combining various immunotherapies like ipilimumab, the CTLA-4 antibody, or drugs that target other checkpoint inhibitors or checkpoint blockade targets like PD-1 or PD-L1 with various small molecule drugs. And a lot of these are in melanoma, looking at drugs that block the MAP kinase pathway that Dr. Chi was talking about. We're looking at this in prostate cancer, where we're combining it with hormone therapies. In the hematologic malignancies, we're also looking at it with this trial of nivolumab and dasatinib. And this trial that we're running here of dasatinib and ipilimumab is really the first time we're trying this concept in sarcoma patients. And the reason why we're doing that is really based on work by Dr. De Matteo, who can talk about this much more eloquently than I can. But really, what he showed was that the efficacy of KIT inhibition with a drug like Gleevec, it's really dependent on an intact immune system. If you don't have an immune system that works, then the Gleevec isn't going to work. And so here, I'll just briefly show you here. If you treat, this is using that GIST mouse model that we've been talking about. But if you treat the mouse with Gleevec, you get tumor over time. That's what you expect. But this was just one of the many experiments in that paper, where what they did was they knocked out components of the immune system and then treated the mice with Gleevec and saw what happened. And what you can see is here, this is the tumor that just grows without any sort of treatment. But this is the tumor if you treat with Gleevec. So you can see that it shrinks from here to here. And then if you knock out several of the components of the immune system, there's really no difference. The tumor still shrinks. But if you knock out the CD8 cells, the tumor doesn't shrink. So that suggests that you need these, you need the functional CD8 cells for the Gleevec to work. And if you look at the tumor, after you treat it with Gleevec, you actually see an increase in the number of good T cells, also suggesting that these T cells are important. And a lot of work went into figuring out, what happened? Why is the response to Gleevec so dependent on the immune system? And it turns out that the answer is because Gleevec, part of the way that it works is because it suppresses a protein called IDO. And IDO is an enzyme that catalyzes the breakdown of something called tryptophan. And when it does that, it breaks into metabolites that are toxic to the immune system. And so he shows that with Gleevec, you actually decrease the amount of IDO expression, both by RNA as well as by protein, and that's what's shown there. So, the next question was, well, can we combine KIT inhibition with a drug like Gleevec with something like CTLA-4 inhibition with ipilimumab? What happens if we do that? And so in this set of experiments, you can see that if you treat the mouse with Gleevec alone, you get some tumor shrinkage for a while, but the tumor grows. If you treat it with CTLA-4 inhibition, which is kind of like what ipilimumab is, you get a little bit of shrinkage actually, but again, it grows. But if you use both in combination, you get better tumor control. And so there's a synergistic effect of ipilimumab or CTLA-4 inhibition and KIT inhibition, at least in this mouse model. And you saw the same thing if you use a drug called dasatinib, which also blocks KIT. And so that was the rationale for this trial, where for the first time we're combining this drug called dasatinib, again, another targeted therapy that inhibits KIT, with ipilimumab. And again, this is a very, very complicated trial where we're actually trying to figure out how can we do this safely. So that's what we call a phase 1 trial, where we slowly increase the doses of drugs until we figure out what the right dose is. And as with all the trials that we're doing here, we're really trying to learn as much as possible. And so that's why we subject our poor patients to all of these biopsies and all these blood tests. They're very, very important, because without that, we actually don't know if we're doing what we think we're doing. And so on this trial, patients are actually getting 3 biopsies. They get a biopsy before they start treatment. They get a biopsy after 1 week of dasatinib, so we know what the dasatinib does to the tumor. And then they get a biopsy after a few weeks of combination therapy, so we can see what the combination actually adds. So that information is very, very important. When figuring out, is this working or not, I think it's really important to remember how immunotherapy works. Because it's not like targeted therapies where, if it's going to work, it works right away and you'll see tumor shrinkage. And so in melanoma, where ipilimumab is approved, this is just a scheme of actually how we treat patients. And it's actually a little bit nerve-wracking, because what happens is ipilimumab is an IV drug. You get it once every 3 weeks, and we do 4 doses. So in practice, what we do is we get a scan. The patients get their 4 doses of drug over the course of about 10 weeks. And then at week 12, which is 3 months after we start, that's when we get our first scan. And we tell our patients at that time that it's very likely that the scan's going to show that the cancer looks bigger. And that's because it takes so long for that therapy to work sometimes. And as long as the patient is clinically doing okay, then we'll just do another scan a month or so later. And that's where we want to see some stabilization or shrinkage. So it really takes us about 4 months or so to know if ipilimumab by itself is helping. So it's very, very nerve-wracking. And so this is what we see in some patients with melanoma. This is a CAT scan, and this is liver. This looks actually really normal up here, although there's small, small spots that I don't think project very well of cancer. And so this patient got ipilimumab, the 4 doses, and at week 12, this is what the scan looks like. And you can see all those spots there. That's all cancer. And if this was, let's say, just in a patient on Gleevec, there's no way we would continue the Gleevec there, because that would tell us that it's not working. But in this case, the patient was actually feeling very, very well. And 2 months later, without any further therapy, the liver looks like this. So most of those spots went away. By week 36, it's even better. And this patient was treated in 2006. He's still doing well, cancer-free, after just 4 doses of ipilimumab. So when we think about, at least in this trial, are patients doing okay or not, we have to remember this kind of kinetics of response, which is unusual. And so on this trial, we've tested kind of 2 dose levels of treatment. And so from the first dose level, these are the patients that we treated, and they were all GIST. And we did that because that's where the science was. That's really why we developed this trial. And you can see that these patients had a lot of treatments before. I mean, some of these patients had, 4, 5, 9 lines of therapy before, and they all had complex genetics as well, consistent with a resistant tumor. And what you can see here is not all of them, but some of them did really, really well. These are patients who have, had imatinib and sunitinib and sorafenib and regorafenib and other drugs, and then progressed. And These patients were on for 24 weeks, so like 6 months, 7 months, even longer. So some of these patients did very, very well. At the next dose level, we wanted to see if we get that sort of level of activity with other sarcoma subtypes. And we didn't see great activity in a lot of these other cancers. This one particular patient was on treatment for a while. But in general, the other sarcoma subtypes didn't do that well. But what was interesting is that we did see that pattern of response where you see kind of initial growth and stabilization, like we saw in the melanoma. And so this is just one example of that. This is a patient with just a 48-year-old gentleman treated with dasatinib at a dose of 70 and ipilimumab, had an exon 9 mutation, and this patient had 4 prior lines of therapy. And what you can kind of see here is us tracking the tumor growth over time. And at this red line, you can think of this as like time 0. And before this patient started treatment, between week -8 and baseline, the tumor growth was about 46%. Over the course of the next 12 weeks after starting treatment, it grew another 38%. But you can see that after week 12, there was a flattening of that rate of growth. And we see that in this patient also. This is a 64-year-old gentleman, exon 9 mutation, a lot of prior therapy. And then we see the same pattern. Between week -16 and baseline, it grew 14%. Between week 0 and week 12, it grew 10%, and then it flattened out. And we saw that again with this patient, 54-year-old gentleman, 2 KIT mutations in exon 11, a lot of prior treatments. And here, between week -17 and baseline, the tumor grew about 26%. From week 0 to around week 12, about 25%, and then it flattens out. So again, I think in some of these cases, we are seeing these immunologic patterns of response to therapy, which I think is very, very promising. And so what we're doing now on the next group of patients is, and I was just talking to Ron, we've had 6 patients upon whom we've actually done those serial biopsies. And so now what we need to do is actually look at those biopsies, interrogate them, see, are we actually suppressing IDO, that protein that we think we're doing? We have to see what else we're doing to the tumor, and then figure out our next steps. Is this something that's worth taking forward? Are we doing it the right way? Do we have to change the schedule? I mean, I do think this is promising, but we just have to optimize that. And so things that we've learned is, one, we've figured out what the right doses are. We think if we're going to give ipilimumab, this actually what's really a high dose actually, then the dasatinib dose that we have to use is 70 milligrams daily. I didn't talk about side effects, but there are side effects that we have to figure out how to manage. But I do think that the tumor growth kinetics are consistent with an immunologic response, and we see that in 4 out of 8 patients with GIST and 1 of 5 patients with non-GIST sarcomas, and we saw CHOI responses as well. And so again, we are in that expansion cohort now where we're doing the biopsies. We've done biopsies on 6 patients. I want to look at those and then figure out what our next steps are going to be with this trial. And because we, again, may have to change the way we're doing this if we're not doing to the tumor what we think we need to do. And so I just want to kind of close with a statement that, there are all these checkpoints that are important in kind of regulating the immune response. And there are drugs now that are being developed for all of these. And some of these drugs look potentially better than ipilimumab. And one of the big targets is antibodies targeting what's called PD-1 or programmed death 1. And so we're working on getting that drug for sarcoma patients as well as some of these other drugs. These other antibodies kind of affect other parts of the immune system where ipilimumab affects kind of the periphery at the level of the antigen presentation between that dendritic cell and the T cell. Things like PD-1 affects, we think, the tumor microenvironment, the interaction between the T cell, for instance, and the tumor cell itself. So again, I think I'm going to leave it there. I do want to thank all my colleagues here. This is a huge team effort, and I think it's a really great team to work with. And I think we can't do this without you guys. This is this is a lot of fun. I think we're making a lot of progress, and we will cure this. We will. Thank you.
So I'm going to tell you a little bit about the clinical side and a couple slides about the lab side. So I'm not really running any clinical trials anymore, but I ran a few that started about 10 or so years ago, and we're still getting lots of data. So this is the longest amount of data for the adjuvant setting. So the adjuvant means you had a single tumor, it wasn't spread anywhere, just a single tumor, you got an operation, and then we gave you a year of imatinib. And what's remarkable on this is we now have a median of close to 8 years of follow-up. This trial had, as you can see, about 700 patients on it. And this was at a couple hundred sites across the country. And what we've proven now, which isn't what we set out to prove, and that's how research goes most of the time, we thought by giving patients a year of imatinib after their surgery that we would cure a certain percentage of them. So if you have a colon cancer and you take chemotherapy, there'll be a group of patients that are cured by taking that chemotherapy, as opposed to not taking the chemotherapy at all. So you can look at this and see, this is the placebo arm. This was a double-blind randomized trial. So half the patients got placebo, and this is how they did. And here's the patients who got imatinib. And the fact that the two curves have come together tell us that, for the most part, nobody was cured on this trial by taking 1 year of imatinib. What you can see, so this is, we're looking at recurrence, so nobody had recurrence the day we started. You know, in the first couple years, the patients who took 1 year of imatinib did much better in terms of recurrence. So, you know, we did this trial hoping that we'd see a percentage of patients that were cured. It turns out that's not really what we're seeing at all, and that's why we got plenty more to do. The best chance of curing someone, of course, is when they just have microscopic disease. So these are patients who had their tumor removed, and at most, they had a couple cells somewhere in their body. The other thing you can see from this trial is that about 75% of patients with that initial tumor without any spread are cured by surgery alone. And we never really knew what that number was, and this is a huge population of patients, and so now we know, you know, what surgery really does. So really, the patients who need imatinib, at least in the invisible disease setting, is just this group right here. So we need to be able to identify those patients a little better. Now, the rescue effect is so great by imatinib that even once you develop a recurrence, and when you do, the tumor is usually very small because we're scanning the patients now at very regular intervals, as you know, as opposed to prior to the year 2000, you didn't really get followed very closely. So even when a patient recurs, we can rescue them very quickly by just putting them back on imatinib. So the patients who recurred on placebo, which you saw was about 25% of the patients, almost a very small proportion of them went on to die, at least so far, because they're rescued by imatinib, and then if imatinib fails, they get, you know, other drugs. So no difference in overall survival by taking 1 year of imatinib after surgery. The only trial that we're running at this point is with a Novartis-sponsored trial. It's called PERSIST-5, and I'm not going to go into a lot of details here, but you might know that the Europeans, after they saw our trial, said, well, we'll do 3 years, and we guessed that 3 years would be better than 1 year, which it was, at least in terms of recurrence. RFS is recurrence-free survival, so the patients who got 3 years had lower recurrence rates, at least while they were on the drug, than the patients who got 1 year. And expecting that, we said, well, why don't we just do a 5-year? And some of the people in this room are people that you would know through the GCRF have been on as an adjuvant postoperatively the drug for 10, or I have patients now up to 14 years where we don't want to stop the drug because you know, we think their chance of recurrence is so high. So I said we have to try to identify who that 25% of patients are who should even receive the drug after that first operation. And some of you have probably heard we've made this with our statisticians. We made this mathematical equation to predict the likelihood of somebody developing a recurrence. And this is online, and I'm not going to go through it exactly, except just to tell you that the score is based on the tumor size, how active it is under the microscope, called the mitotic index, and where it arose. And so we can take a patient that we've just operated on and say, well, we have a rough idea of the likelihood of recurrence in the next 2 to 5 years, and that helps guide us as to whether they should take the drug or not. So the other piece of information that we have is that Christina and another pathologist took all these patients. We had 700 patients on this trial. It's a huge trial. And we got tumor specimens on about 550 of them, or 600, and they ran the mutation analysis. So we have a lot of information now about how patients with different mutations did in relation to either being on the placebo arm, not getting the drug up front, or getting imatinib. And you know, if you look at our nomogram that I just showed you, you might say, well, gee, where's mutation on there? And again, many times you're absolutely wrong in research. So we assumed that once we got all the mutation data, that we would make a better nomogram. It turns out the mutation data doesn't help the nomogram at all. And what I'm showing you here is a way of mathematically representing what's important in the nomogram or the prediction. And so tumor size, important. So if something crosses this line of 1, it has no relevance. If something makes things worse, it's to this side, and makes things better, it's to this side. So a big tumor, well, as you would imagine, those patients have a higher likelihood of recurrence. Small bowel location, higher likelihood of recurrence. This has been known for a very long time. That's why we made our nomogram using these parameters. High mitotic rate, same thing. This is the most important determinant of recurrence. But then when we start throwing in whether the patients had exon 9, exon 11, PDGFR, no difference. So what that tells me is that this information that we thought was going to be so important is somehow just contained in mitotic index location and tumor size. So our nomogram is still the most accurate way of predicting the likelihood of recurrence, and we don't really need to add mutation to it because it's not adding anything. So you know, we started out saying, well, let's try to cure the patients with primary disease with just the single tumor, and you know, we have lots of other ideas like adding immunotherapy up front. We think that might be where the biggest impact of immunotherapy is, and hopefully we'll get to that at some point. But we started thinking of other things that we could do. And so I'm going to give you another example where we had this great hypothesis. It made so much sense. We said it will definitely work in the laboratory. And it worked, but for none of the reasons that we expected. So we said, let's take this new compound that was being made by this company called Plexicon. And we found this compound several years ago. It inhibits KIT, so it's like imatinib. But in addition, it heavily inhibits this other receptor that sits on the cell surface. And this receptor in particular sits on the surface of an immune cell called a macrophage. And macrophages turn out to be the most common immune cells in most human tumors. And you can think of macrophages as kind of like little Pac-Men. They go around and eat things all day. They eat dead cells. They can help eat tumor cells. They can help with infections. And so this made perfect sense because it was well known at the time, or at least well conceived at the time, that macrophages provide incredible support to tumors. They do all kinds of things. They help grow blood vessels, and they release cytokines. And so this made perfect sense. Let's inhibit KIT, which will knock down the tumor cell, and let's inhibit the macrophages and get rid of the macrophages, and then the tumor will be paralyzed in 2 different ways. And so we gave this drug to our mouse model, like you've heard about several times. And even just after a short period of time, we measured the tumor weight, and the control, this is really no treatment, had that amount of weight. And we gave imatinib, and this is what we typically see. The weight drops by about 2/3. And we gave this drug, and we said, oh, well, okay, that's okay. I mean, it seems a little better, but when you see an effect that small, you might repeat the experiment, and that effect is gone the next time. But when we start counting the number of tumor cells, the KIT-positive tumor cells, there was a dramatic difference. And you know, when we went a little further and said, well, let's look under the microscope, then it became extremely clear that it was night and day. So here's the control tumor tumor cells here, and what we're staining for, Ping showed you a stain like this. The blue is kind of scar tissue. Think of it that way. And it's just a low magnification and a high magnification. So within a normal tumor, there's very little sort of scar tissue. It's just helping hold the tumor together. If you give imatinib, yeah, maybe there's a little more blue. It's not dramatic. We give this drug, and there's so many fewer cells. It's incredible, and there's this huge scar response. And it turns out this is what we see in human beings also, using imatinib alone. If you treat them for many, many months, you'll start seeing all this scar tissue. And then if we just stain for KIT, which marks sort of the active tumor cells, you could look at it that way. This is a little small, but there's plenty of brown staining, which is the KIT staining. You can see imatinib, plenty of brown staining. And here, there's hardly any brown staining. And this is at many, many weeks. These are just at 4 weeks. But when we use this drug for 17 weeks, the tumor is nearly obliterated. We haven't cured the mice, but we've come the closest to cure with this drug compared to anything else that we've tried in the laboratory. Okay, well, that's nice. That's our mouse model that develops the tumor on its own because of this mutation that's been engineered by the Besmer lab. But what if we take human tumor cell lines, because that's the other way we study cancer, and we have special mice that have no immune system, so you can put a human tumor into them, and the tumor will grow. And so this is one tumor line here, where we let all the tumors grow here, and then we randomize the mice to either get control, which is nothing, or just like saline injections, or we had imatinib here, and this particular cell line is stable on imatinib. It's not decreasing in size at all, at least in the mouse. And then here we see a dramatic. Even within 2 weeks, this tumor has been decimated. And here's the actual examples of some of the tumors. Big, moderate. This is the size that this was at the beginning of the experiment. They were all about this size or just slightly smaller. And there's just a little remnant of this tumor. And this is only after 2 weeks of therapy. So pretty dramatic findings. And then same thing, we look at tumor weight, and okay, now we got a bigger tumor weight difference in the human cell lines. But when we try to count KIT-positive cells, there's hardly any KIT-positive cells. There's still a few there. And if we had treated for much longer times, maybe they would have gone away. But this is we don't see differences that often in the laboratory with a drug like this. So we're very interested in bringing this drug to humans, and we've actually identified another drug that is more commercially available already, because this one is largely still in clinical trials, but we found now other things that can behave in a similar fashion as this drug. So we're hoping to bring new drugs. We started out thinking this was all going to be about the immune system because it knocks out the macrophages, and I'm not showing you that data, but it depletes the macrophages within the tumor very well, and yet it's having this great effect. So that we're still kind of puzzled by this, like how could there be such a great effect when our hypothesis was completely wrong? And this is the last slide I'm going to show you. And it's really not any data. It's just showing you the overall concept of what we published a few months ago. And so I told you that everybody had this sort of conception that macrophages were very supportive for tumors. And all that data really came from 100 different experiments that were done in mice. But nobody really looked in detail in human beings. And being at this institution, and for patients who consent, we can get fresh tumor specimens, and we can take them right to the laboratory and pull the macrophages out and study their function and their properties. And what we found was, at least so far in this tumor, we're going to look in other human cancers, but in the mouse and in the humans, the macrophages are behaving exactly opposite to what the entire immunology field was believing. We actually find that the macrophages are at baseline what we call M1. M1 means they're anti-tumoral. They're very inflammatory. And they like to stimulate T cells. T cells are what can also kill tumors. And so this was very striking. In the mouse and in our human patients, we found that macrophages at baseline are M1, completely the reverse of what the rest of the world thought. Interestingly, in the mouse and in humans, when we give imatinib, they flip to M2, which are more tumor supporting, which is what, at least in all the other mouse models that are out there, the cells are like. And the M2 cells actually suppress sort of anti-tumor properties. And we found sort of how the mechanism of this is working, but very striking. And then we haven't looked in Benedict's mice yet, but we'd like to at some point. But in the human beings that become eventually resistant to imatinib, what we find is they go back to this M1 state. And it doesn't make sense that a tumor would want inflammatory macrophages around it. So we're trying to figure out most things in biology, there's usually some advantage, whether it's immediately apparent, it may not be, but why would a tumor want to surround itself with macrophages that are actually in some ways acting against it? And we haven't figured that out yet, but that's something that we certainly want to figure out. And this also, from a therapy point of view, means if they're already anti-tumoral, what can we do to stimulate them to push them to be even more anti-tumoral? So this is some of the things that we're working on. So obviously we're very appreciative of the GIST Cancer Research Fund, and this was the group that really allowed us to start doing the immune experiments that we started probably now quite some time ago, maybe about 6 years ago. And I brought the lab so you could see who they are. Maybe the lab could stand up, and we don't have to have an interview over each one of them because it's at the end of the day. But nearly all of them are working on GIST. So we hate GIST as much as you do, and we're going to continue to try to develop new ways to treat it. Thanks very much.
Okay.
Anyway, it's been more difficult to raise money this year. We actually received 20% more donations and 20% less money this year. So it's because of the economy and everything else. We need more help. We don't need more donations dollar-wise. We need more people doing events like Eleanor did last Monday night in Long Island. Dr. DiMatteo, Dr. Antonesco, they both drove out there over 2 hours after they worked here to the event and everything else, which we really appreciate.
2 years in a row.
2 years in a row. Anyway, to make a long story short, I also want to commend you guys in the back for all the work you've been doing. I also got to show you one thing to get a little laugh here anyway, okay? Last year, they sent me pictures of the event here. Well, here's a picture I blew up of Christina. I gave her a check, and look what she did. She held it in front of my face. You can't even see who I am. Here. We raise all this money for this institution, and that's what I get in return. And then they mailed it to me yet. Here, Christine, you can hang this up in your house. But, seriously, it's been very difficult, this year raising money. I had a whole bunch of other things to say. It's not easy to do. We don't receive any salaries or anything else. It's all volunteer. We have raised over, in 13 years or 12 years, over $7 million. We have given this institution, it's close to $1.5 million. And every penny that we raise goes to just research. Nothing goes to salaries, nothing goes to anything else. The only help that we get from Novartis and Pfizer, and they help share the expenses to operate the organization, and that's basically it. So now I want all you guys that work in the different labs, not only for Ron, I'm sure some of you are from Christina's, from Dr. Ping's. Come on up here for a minute. Just come on up here. You guys deserve the credit too. These guys get all the credit. You guys deserve the credit. Come on up here. Come on. This way you get into a picture. You don't have to be embarrassed. Let's go. Come on. No, come up here. Come up here, right up here. Let's go. Come on, you guys do all that work and everything else. You deserve a little credit. Come on, walk. Come on, up here. Look how slow they move. Now, okay, Eleanor, come on up here. Mark, please come up too. Come on. Tanya, come up. Ron, come up. Now, I'm not only presenting this to the doctors, I'm presenting it to you guys because you guys do probably a lot of the work. Okay. It's a little bit less than last year, but for Dr. DiMatteo's lab, here's $100,000. For Christina's lab, here's $100,000. But listen to me.
Can you get a picture?
I'm not standing next to her this year either. But we need all the help we can get, and hopefully we don't have to come back next year because these guys right here are going to find the cure. So no more vacations. You have to work on weekends and everything else.
Wait, we don't give them vacation already.
Oh, okay. Anyway, I'm finished talking, and thank you all. Thank you all for your support, and thank you for coming here today.